Today, and yes, we are very delighted to present you with a session entitled Galaxy Global Platform for Analysis of SARS CoV-2 Data and Variants. My name is Idris Suleiman. I'm from the e-Research Network for Southeast Asia, and we're very proud to be able to introduce you two of the very most qualified trainers from Australia. We have Dr. Gareth Price, who is a genomics scientist at Queensland Cyber Infrastructure Foundation Limited. He has over 15 years of experience in running laboratories utilizing microarrays, NGS technologies to address a variety of human and human pathogen-related conditions. He's been involved in experimental design, assay performance, data quality control, data analysis, data interpretation using omics technologies. And he's today joined by Dr. Ann Sim, who is from the combined Biocommons, Galaxy Australia, Melbourne Bioinformatics team. She has been working on deploying complex workflows to Galaxy and has had a wide variety of bioinformatics, life sciences knowledge, which she's hoping to demonstrate today. She's based at the University of Melbourne in Australia. But just before we start, I must say that I come from the technology policy perspective and would just like to allude to a very interesting study that I've discovered recently, which shows a comparative analysis of how different countries perform. And I found some very, very interesting contrasting information, which shows that there isn't really a correlation between countries' high income per capita and the amount of genomic sequencing efforts, if you like. So just quickly wanted to show you this study, which is one that has been produced by Dr. Yuki Furuse. It's in the International Journal of Infectious Disease. And the title of the study is Genomic Sequencing Effort for SARS-CoV-2 by Country During the Pandemic. And the most interesting aspect is that, you expect that certain countries like the UK, USA, Australia, Iceland have performed high proportion of samples that have been sequenced. And this is also the case in Iceland, Netherlands, and most of the other countries, including Australia. However, three countries stand out, and these are the Democratic Republic of Congo, Bangladesh, and Malaysia. So yes, and out of those three, you'd expect perhaps the DRC may not actually been able to produce as much sequences or a high percentage of sequence per case, but they do. I mean, like more or less the DRC and Bangladesh is actually in the middle of the graph that I'm showing you there. And then if you look at sort of more granular measures like fraction, timeliness, and openness, you find that DRC is really nearly on top of the table, just behind Australia, and Bangladesh a little bit further down, and Malaysia also at there. But there don't seem to be too many other countries except Thailand, maybe also in the middle, which I'm sorry I didn't put it in there. But Thailand didn't appear in the first place. So basically, I like to showcase this to just show that perhaps, there is a lot of effort that might be required to strengthen further sequencing capacity at the global level, which will help in the fight against this pandemic, but not only the current pandemic, but also maybe in the future outbreaks of viral diseases. So yeah, I just wanted to allude you to that study because I found that it really is interesting variety across different countries. So without further ado, I wanted to now hand over the floor to Dr. Price. Gareth, please take it away.
Well, thank you, Idris, and thank you to the organisers and the attendees for allowing Anna and myself to present over the next 3 sessions. We've taken on a somewhat ambitious task, but one that I think is very important. Idris has laid the groundwork for it extremely nicely with those 2 slides there, and that is to give you a grounding in the activity that Anna and I spend much of our time in, which is a platform and a service called Galaxy that you'll get to know very well as we talk today. And in the second and third session, how the global Galaxy project, which again, we'll highlight what that is, came together to build infrastructure, tools, and analytical pipelines or workflows for analyzing SARS-CoV-2 data and trying to make that a uniform and accessible process globally. So we're going to have 3 sessions today. The first session is scheduled for 1.5 hours. It probably will not go that long. The next 2 sessions are probably going to be closer to time. From a practical point of view, we will be monitoring the Zoom chat and also the Hoover app. So please do post questions you have in either of those locations, and we will get to them as we can throughout the day. This first session is going to be grounding you in what is Galaxy. I think you need to understand the place that we'll be working in and the kinds of things we'll be showing you in terms of data before we can move into the more complex part of it, which is how to analyze SARS-CoV-2 information inside that platform. So we're going to go through what is Galaxy, understand how and why it's deployed. We're going to look at, actually, Anna will take over at some point soon, and we'll be looking at how to display a file, work in Galaxy. We've got a bit of echo from someone. If you're attending, could you please mute your microphone unless you have a burning question, and please do feel free to use the Zoom functionality to raise your hand or interrupt if you have something you'd like to ask. So, the biggest question, the one that we're advocating for, what is Galaxy? And I'm going to go through a variety of slides to answer this question. And you're going to see it very shortly too. But at a very high level, Galaxy is a web interface. So it's a website to a complex mixture of computing resources. And you can have almost anything behind that. It's a launching site for analysis tools and many tools with predetermined and scalable resources key to a user's data set size. So what does that mean? Well, in very practical terms, what it means is we have pre-configured the amount of compute that will be launched when you bring a data set to Galaxy. Bring a small data set, you get a proportional amount of CPUs. Bring a very large data set, you get a much larger allocation of CPUs to analyze that data. And our experience of operating a bioinformatics service and what we know from our global colleagues is one of the biggest challenges that users face when they interact with high performance compute or HPC environments is estimating the resource time and therefore the wall time of their jobs. And this is something that Galaxy takes on for you. A Galaxy is also, really importantly, a repository for all the reference data you need or require for your analysis. And the subtlety in that is it's already pre-computed to run as quickly as possible. It's not just a tool platform though, it is a workflow platform, and that's one of the proudest and most robust and used features. So you can create and execute workflows in a predictable and reproducible way. And finally, it is a data and results sharing platform. So not in the way that Dropbox, for example, or your AWS bucket is. It's not for passing through data, but it's for bringing your data in, doing an analysis and sharing that analysis with your colleague or your supervisor or whoever it is, sharing the results, the methodology used, and the results of that workflow. So we, Anna and myself, both operate Galaxy Australia, which is one of the three pillar stones of a global use Galaxy servers or ecosystem, as we like to call it. So this just really iterates what Galaxy is, so I can talk to it for the next few slides. It's a website. On the left-hand side is a list of tools for your analytical options. On the right-hand side is your history. And a history is a place that you bring your data, you compute inside that history, you generate results in that history. And each history can be key to a particular data set, a particular experimental methodology, or a trial and error that you're going through to understand various algorithmic tools you might need. And the middle window toggles between information tools to contextualize Galaxy. So we've done what is Galaxy, and I'm just going to disable my video just to make sure that I have the bandwidth to continue to present. I am based here in Queensland in Australia, and we have just had a fairly large flooding event that has affected everyone's internet, so apologies for that. So where is Galaxy? Well, Galaxy is all around the world. It's been in operation for many years, approximately 15 years, but this graph starts at about 2013. And it shows all the public Galaxy servers. So these are places that you can come to and perform your analysis almost without condition. Now, every Galaxy server has a condition, but these are public servers in the sense you can discover them, find them, and analyze data. And I think we used to say there's about 120, but we're creeping up to that 150 mark. And we also have various other ways of deploying Galaxy that are somewhat newer and starting to trend upwards. So where can you access Galaxy? And this is all the things that we'll be doing, or you'll be following along or watching today. Well, there's the public servers, and I've just mentioned them, and there's a ton of those. These are all found at galaxyproject.org/use. We'll show you this list of 120 servers. It is possible to spin up your own Galaxy on your laptop, on the cloud, or on your organization's own infrastructure. And they all come with various levels of complexity, but they also come with various levels of two very important things. One, customization of tools, and two, the ability to keep any data that you perceive as sensitive behind your firewall. So probably the most popular ways we know people access Galaxy are public servers on the web and in their organization. And this last one is about generally analyzing human or human-related data where your organization has a policy not to share that on the open web. Therefore, you hide it behind your firewall. And like I said, we make best estimates to how many of these. We know of hundreds, but there could well be thousands because that's the nature of them. We can't see them behind those firewalls. So we've done what, we've done where. Now, why is Galaxy? Well, why is to enable science and to enable researchers to access the best analytical tools to get them through their data analysis quicker than they might do by trying to access HPC, cheaper than they might do by accessing commercial bioinformatics software. So therefore, one of the best ways, if we're using science as our metric for why is Galaxy, is to track publications. And either cumulative publications as shown on the left-hand side of the screen, or publications by year shown on the right-hand side of the screen, shows that from the early days of Galaxy through to the last couple of years, there has been a consistent trend and growth in the amount of activity that Galaxy has enabled. And it was with no small amount of pride that Galaxy Global celebrated this milestone here on the left graph, which was 10,000 peer-reviewed publications in mid-2019. So I think a wonderful example of how this service has enabled a huge amount of global science. And the last one is an important one, and this is one that we hopefully will leave you with a sense today that sometimes when you're working in the lab, or certainly in a bioinformatician working in front of your computer, you can feel a little bit isolated. So who is Galaxy is a great question, and Galaxy really is the community. So I have put a picture in here of one of the founding principal investigators of Galaxy, James Taylor, who sadly passed away 2 years ago. But he was a massive advocate for the fact that Galaxy is about community. And this is a photo from the last time we were fortunate enough to meet at our annual conference before COVID came crashing down. So this was the 2019 conference in Freiburg in Germany. And we're looking forward to meeting again this year in America. So I hope by the end of our sessions today, we've convinced you to become part of this Galaxy community, contribute your analyses, your tools, your data to making basically global science a bit better. So that's the very high level version of Galaxy. I'll get a little bit granular and then we'll be passing over to Anna to take you through some of the practicalities of how it works. So I'll be addressing this shortly, but there are thousands of tools that Galaxy can operate. But running one tool is a bit like watching a kettle boil. If you need to run 30 tools, you're effectively watching 30 kettles boil, but you can only switch the second one off when the first one finishes and so on and so forth. So it's a very laborious and inefficient task. So Galaxy really gets its power through being able to build workflows. And this is a workflow of about 30 different tools here that are actually taking a metagenomic sample, separating out the various bacterial types, counting them, and ultimately plotting them in a digestible graph format. And all this workflow can be managed by a single play button. You load up all your data, everything runs, it's all scheduled for you, it's resourced appropriately. You can share this workflow with your colleagues and you can run it over and over again. And this is really to the heart of what makes Galaxy such an efficient platform. And we'll return to this concept when looking at SARS-CoV-2 analysis and where workflows helped take the burden away from the user and put it back on the computation to process the data. One of the we'll get to at the end of the day is now that those workflows turn this workflow through markdown language into a graphical report. So again, if you're the bioinformatician in your group, if you're the genomic scientist in your group, these relatively digestible workflow reports, and share that with your colleagues, and that becomes their entry level to the results that are being generated. I just alluded to the fact that there are many tools in Galaxy. So here's another graph. Valid tools, so operational tools and the authors of those tools, tracking from 2015 through to essentially the start of this year. And you can see there's a consistent, again, upward trend in data. And I checked the website the other day, there is now 8,591 different analytical tools available to be deployed on Galaxy. And these don't just cover proteomics anymore, genomics anymore. They cover proteomics, cheminformatics, ecology, artificial intelligence, more and more imaging, and we're obviously hoping with time to cover all the domains within the life science and then beyond life science. What's not shown on the graph because it's not really as easily graphable, but again, Galaxy hosts hundreds of pre-computed references for rapid analysis as well. I mentioned, I think on almost my very first slide, that Galaxy is a simple web portal to a complex compute resources. Currently in Australia, for Galaxy Australia, which is found at usegalaxy.org.au. If you're accessing this website, you're accessing what we call the head node here at a supercompute facility in Western Australia, But in fact, we're actually running your jobs all over the country. So we're running them at the University of Melbourne. We're running them at our national compute infrastructure in our capital city in Canberra, and we're running them here in Brisbane at my organisation, QSIF. Each of these blue dots can be configured with different capacity and style of CPU and RAM and storage to really tailor the compute to the styles of questions you're asking. And that's what I mean by a complex compute backend. And I think I'm nearly at the end of my section, but one of the important places to keep in mind and highlight, and I'll put the web link into Zoom chat shortly and into Hoover, is the Galaxy Training Network. So we've talked, or I've shown you, hopefully very briefly, that Galaxy is about workflows, it's about tools, it's about reproducible data. And one of the consequences of that is that you can write some really fantastic, rich biological and informative tutorials on how to do elements of biology and make them available to others. So the Galaxy Training Network is a website of different training topics covering aspects of biology and statistics and proteomics, where you get deep follow-along guides for how to process data in Galaxy and what Galaxy is set up for SARS-CoV-2. But if you are interested in a new domain of life science, then coming to the Galaxy Training Network is a fantastic place to learn about how to process that style of data. In a particular topic, and I've just picked single cell, you'll get introductions, processing, results, a slide deck, a follow along. What's hidden behind here are some additional features, but you get all the data because it's publicly available, the workflows, and in this final column here, where around the world it could be run, on which public Galaxy server. Which brings me very nicely to talk about this particular event that's coming up next week, which is the Galaxy Training Network Smorgasbord #2, otherwise known as the Galaxy Training Network Tapas event. This is a week-long asynchronous global training event where you can come, you can find any number of these tutorials, follow along with guides, but with an open Slack channel to communicate with the authors of those guides or tutorial experts who can help you walk through those different biological topics to learn again about how to use Galaxy to perform these analyses. My colleague Anna, who will be presenting shortly, is one of those people that will be manning this event next week to make sure it goes smoothly. So, before we jump into the basic training, some context. So, I'm not going to spend time convincing anyone on this call that the analysis of SARS-CoV-2 is an important thing to do. I'll take that as given. But what wasn't given was in the early days of analysis of COVID-19 data or SARS-CoV-2 data. And everyone was scrambling to find what they believed were positive samples, looking to analyze them as quickly as possible and make that data publicly available, which was actually fantastic because the more data was out there, the more we understood. The principal investigators of Galaxy, and there are a number of listed through here, their personal passion was viral genomics. And when they looked at the data that was being produced globally, they could see that one of the core problems, and I see this both humbly and bluntly, one of the core problems in those early analyses was not a consistently applied methodology to how the analysis was done. So very quickly, a series of workflows covering genomics, evolution, chemoinformatics, proteomics, ARCTIC, which is amplicon-based analysis, and RNA-seq was produced by Global Galaxy and rolled out across Galaxy Main, which is America, Galaxy Europe, based in Germany, and Galaxy Australia. This was published and then matured into this more recent Nature Biotech correspondence here, talking about how the Global Galaxy e-research infrastructure has matured to support COVID or SARS-CoV-2 monitoring and analysis. And we will be spending session 2 and 3 looking at the resources on this particular page here, the COVID-19 at Galaxy Project or .galaxyproject.org. So with that, I'm going to hand over to Anna. She's going to take you through interactions with Galaxy. For those of you that wish to follow along, we acknowledge that the speed we might be going at today might be a bit faster than we would typically do for an extensive deep training event. The purpose of today's session is to follow along if you want to, but also to give you a really deep understanding and breadth of what Galaxy can enable. And so you can plan your own e-research infrastructure, you can plan your deployments of Galaxy, or you can make use of the pre-existing Galaxy servers to analyze data you have. With that, I'll unshare my screen, hand over running authority to Anna. Like I said, I may have not said, I'll be monitoring chat in Zoom and Hoover, so please do ask any questions you want in each of those forums. Thank you, Anna.
Great, thanks, Gareth. I'll just share my screen. Can't seem to, oh, there we go. Okay, can everyone see that screen okay, showing the Galaxy window and a tutorial?
I can, Anna, thank you very much.
That's great. Okay, so yeah, today we're going to have a short introduction about using the Galaxy platform. If you've used Galaxy before, this may be a bit of a refresher. And if you haven't, hopefully this will be fairly clear and a fairly simple way to start using the platform. So we'll get familiar with how it works, and then in the later workshops today, we can use Galaxy to analyse some SARS-CoV-2 data. So as Gareth mentioned, there are several Galaxy platforms available around the world, and today we're going to use the Galaxy Australia platform. If you'd like to follow along the steps as I do them today, please feel free to do that. And if you'd rather just watch the steps and perhaps try the tutorial later, you're also welcome to do that. So to get started, I'd recommend opening 2 browser windows, like I have here, one with Galaxy Australia and one with the tutorial that we're going to follow. Could I ask you, Gareth, to perhaps paste both of those links into
Absolutely.
the chat? Thank you.
Just be 30 seconds behind you, but I'll do it.
No, that's not a problem. And as Gareth mentioned, this is one of many Galaxy tutorials that are available via the Galaxy Training Network, which is a really excellent resource. So hopefully you've been able to open up one window with Galaxy Australia and another window with the tutorial. So what we're going to do today is a brief overview of the Galaxy interface. We're going to do some really basic steps, which are naming a history, uploading a file, using a tool, looking at the results of that tool, running other tools, and finally looking a bit at the workflow functions in Galaxy. So if you're following along, please look in your Galaxy Australia window now and go along the top bar to User and click on that drop-down arrow. And if you haven't used Galaxy before, or Galaxy Australia, please register as a user. And if you have used Galaxy Australia before, you can log in through here. So I'm just going to wait a minute while people get registered or logged in. And if you're having any issues with those, please pop them into the chat and Gareth or I will try to assist where we can. So while everyone's doing that, I'll give a brief chat about what Galaxy looks like. As Gareth said, the Galaxy window is a window in your browser. So you access it via the internet, and it's a big window. And on the left-hand side, we have the tools, and the right-hand side, we have the history, which is a record of all of the data that you've put into Galaxy and all of the tools that you've run and the results. And all of this is discussed more in this tutorial. So if you find some things go a bit fast or you miss them, you can always refer back to this written tutorial to catch up. So yes, in Galaxy, we have the tools on the left, the history on the right, and in the middle, it's sort of a viewing panel. It's for viewing either some of your results or viewing the tools when you use them. And that will become clearer as we go through this tutorial. So the first time you use Galaxy, there won't be any files here in this history panel. So let's give our history a name. We can do that by clicking on unnamed history, and you can call that whatever you would like. This is not very inventive, but I'll call it today's analysis. That's probably not a good idea. Probably best to call it something very informative and easy to search for. So there's something not to do. So we've named our history. And then we'll go to the next section, which is uploading a file. So as we said, the tools are over here in the left-hand side. And one of the ways to get data into Galaxy is to click on this little box that says upload data. So we'll click on that. And today we are going to get data into Galaxy by pasting the address of a file. So in the tutorial, there's a link here as a Nodo record for a file. So I'll press copy. And then in this Galaxy box here, I'll press paste fetch data. Brings up a window here. And I will paste that in there. Then click start. And then click close. So please let us know if you're having any trouble with that, if you're following along. But what we did was we went to the upload data box in the box. Oh, is someone having trouble? Perhaps paste your questions. Oh, John? Perhaps paste your questions into the chat. That's probably the most efficient way for us to troubleshoot. So we went to upload data and we clicked on that and we pasted in the address of the file. And then now the file should be in our history over here on the right-hand side.
Yeah.
Sorry, I didn't catch that. Perhaps you could mute and write your question into the chat function. Thank you. So we have our file here, which is good. Let's have a look at what this file is. There's this little icon here, which is an eye. So if we click on this, this shows us what the file is here in this center panel. And here we can see that this is a FASTQ file of sequencing reads. There's information in the tutorial here about what a FASTQ format is, if you're not familiar with it. But basically, this is a whole set of sequencing reads with identifiers. That's the identifier, that's the sequencing read, and so on. So we've got our reads into our history. So now we'll use a tool we'll go to the tool panel, and there's a box here that says search tools. We are going to use a tool called FastQC. So I'll type that into the tool panel. And we can see that it's popped up here at the top of the list. We click on that tool. And now here in the center panel, we have the information about this tool where we can tell it the files to use and the settings to run with. So here it says raw read data from your current history. It's automatically found the file that we want to use. So we'll just confirm that, yes. And today we're not changing any other parameters in this tool. So the main thing is we've given it the correct file and we scroll down to the execute button and then we click that. And now this tool is running on that data. And what you can see over here in the history panel is that there are two new files appearing at the top of the panel, which are the results from that tool. And currently on my screen, these files are orange, which means the job's currently running. And when it's finished, if it's been successful, those jobs will turn green. There we go. So we've got two result files here. We will look at the one that says FastQ on Data 1 webpage. I should just note here, with Galaxy in the history, it numbers the files sequentially. So our first file that we input, it called file number 1. And when it ran the tool FastQC, you can see in these results, it said FastQC on Data 1. So it always tries to be quite clear about what it has done on what file. So it's quite a nice way to keep clear what you've done in your analysis. So we will look at the one that says web page, again with the eye icon. And I'm just going to drag or actually collapse these side panels so we can see a bit more detail here. So these are the FastQC results on our input file, which was FastQ reads. And what this tool has done is it's got a lot of information about that file and told us, which is a common thing, I guess, we would all do in any analysis we were doing on sequencing. We often want to know just the basic information about those sequencing reads, how many there were, how long they were, and what sort of quality, sequencing quality they had. So one of the things that FastQC does is it shows us the sequence quality at every position in those sequencing reads. So this is a nice graph that's quite useful. We can see along the bottom, there are 150 base pairs. So that's the lengths of all of our reads. And we can see that best quality is in the center of the reads, and it's fairly good the whole way along. The worst quality is probably at the start of the reads. But overall, these are fairly good quality reads. So I'll just reopen these side panels again. So next we'll run another tool. So that's the first tool we've run. We're going to run a tool now to filter some of these reads. So in the tool search box, I'm going to search for filter by quality. And the topic here is filter by quality. I'll click on that one. And the information about the tool is here again in the center panel. So we want to tell it the correct input FastQ file. This is our initial file that we uploaded. Then we would like to give it a value for the quality. We want these reads to have quite good quality, for example, in this analysis. So we'll say 35. And then we want 80% of the bases in every read to have that quality. So we'll say 80. This is just an example. These would change according to your requirements. Now we will click execute. And again, if this is going a bit fast, my apologies. It's always hard to do virtually where I can't see everyone at their desks. So I'd just like to reiterate that the instructions hopefully are very clear in this tutorial, and they're designed for people to run at their own pace. And there's lots of help information in there. So if you need to run this again after this session, it should be fairly straightforward following the materials. So we've run the tool called Filter by Quality. This has given us one new output file in our history. This is file number 4. So let's have a look at the results. So remember how we set some parameters for how much we wanted the reads to be filtered. We wanted them to be a certain quality over a certain amount of each read. In this case, we can click on the eye icon. I'll do that now. But this won't be super informative because it's just a list of sequencing reads and we don't really know how many have been filtered. So instead, we will just click on the file name itself, and this brings up some metadata about that file. And we can see here our filter settings, and it also tells us how many reads were filtered out. So it discarded almost 1,800 reads. So that's useful information. That's what we want to find out. So perhaps we think, oh, we might actually like slightly better quality reads. We seem to have a lot of reads. Let's change those quality settings. A really useful feature in Galaxy is an ability to rerun jobs quite easily. And to do that, there's a little arrow icon under any of your output files, and you can click on that icon, and that brings up the tool and the settings that you used previously. And here you can alter the settings and rerun the same tool on the same data. So for example, here, we are going to change the quality to something slightly higher, just to 36 from 35. And everything else will leave the same, and we will press execute. And now a new file is going to populate at the top of our history. And that's green, so that's finished. And again, we can click on this file name and look underneath it, and it tells us it has now discarded more than 11,000 reads. So that's quite a big difference. Initially discarded less than 2,000. So that's really good for us to know if we were planning our analysis. So it's a good way to rerun tools and vary the parameters in Galaxy. Another thing we're going to look at today is another neat feature where we can take our history of all the steps that we've run and all of the parameter settings that we used, and we can actually convert that into a workflow that we can reuse again. So the way that we do this is we go to the top right-hand side of the history panel, and there's a little icon called a cogwheel. So I'll click on that. And then we go down and we select extract workflow. And that's bringing up information now about all of these steps that we ran in our current history. So we can make a few changes here. Here for our input data set, we can change this to a bit more of a general name. So we'll call this FastQ reads. And then perhaps we think that second set of filtering that we did, perhaps we prefer not to include that. So I'll uncheck this arrow here, this tick mark. And so then we'll give it a name. Maybe we'll give it, follow what the tutorial says, QC and filtering. And then we'll click create workflow. So that has made the workflow. We'll have a look at that in a minute. First of all, we are going to create a new history. And we do that by going to the top of the history panel and clicking the plus button. Again, I'll give it a very bad name. Next analysis. And that's currently empty. But we might want to look at all of our histories, all of the things we've been doing. And the way we can do that is by going to this, I think this, oh, is there a question? So I think this is a book icon. We'll click on that. And this will show you all the histories that you've created in Galaxy. And you can drag along the bottom bar to scroll. To go back to the home screen, press the little house icon. We'll actually populate that history with a file. So let's go back to looking at all of our histories with the little book icon. And let's drag the original file that we used into that new history. So you'll see your previous analysis in all of your histories and your next analysis, and you can drag any files that you've used previously over into that current history. So I dragged in the FastQ reads into that history. And then press the home button back to the home screen. Now we have one file in this current history. So we're going to run the workflow now in this history. So to do that, we go to workflow, the workflow button in the top of your Galaxy menu bar. And here is the workflow you created, the one called QC and filtering. One thing we might just do here is have a look at the workflow as it exists in a visual form. So by clicking on the drop-down arrow and then clicking edit, we can actually see what this looks like on this is called the workflow canvas. And obviously we don't have a lot in this workflow, but we can see what's in it. We've got our input reads over here, and we send those reads to a tool called FastQC, and we also send them to a filtering tool. So we'll go back to our current history. So in the workflow list, we can see our workflow here called QC and filtering. To run this, we just need to scroll over with this bottom bar. Oops, to find the run button. We click that. And then we've got some options here for the workflow. So we need to check that we've got the right input files. So we do. Our FastQ reads, that's our correct file. We don't need to change anything in this workflow today. So we'll leave everything else as it is, and we'll click Run Workflow. And that's quite a neat way to rerun workflows that you've done for other analyses on new data. So you can just save what you've done previously and rerun it in a new history with all of the tools that you need. So just to give a quick summary of what we did today, we learnt about the Galaxy interface. It's got the tools on the left-hand side, a viewing panel here in the centre, and histories over here at the right-hand side. You can create new histories if you want to do different analyses. If you want to get data into Galaxy, you can do that with the Upload Data button. And there are other ways to get data into Galaxy, and they're discussed more in other Galaxy tutorials. You can choose tools to run, and you can change settings in those tools. The output files from the tools appear at the top of your current history. You can look at the files by clicking on the eye icon, as we did, for example, with our FastQ reads. You can look at all of your histories by going to this little book icon, seeing all of your histories. You can switch to previous histories in here as well. And then the home button to go back home. And if you're using Galaxy while you are logged in, you'll be able to log out of Galaxy and everything will be saved in there, and you can log back in and continue on. So that's the end of the brief introduction to how to use the platform, how to use Galaxy Australia specifically. The instructions are quite similar or really the same for any of the Galaxy instances, but today in the analyses that we'll do later, we'll use Galaxy Australia for some of those. So if anyone's got any questions, or if Gareth, you want to add anything to that, please feel free.
I'll pause for a second, Anna, but then I'll just got one or two summary slides before we close the session. Yes, so I can only encourage people to post your questions as you think about them in the Hoover app or in the Zoom whilst it's still running. And so thank you, Anna. What you'll see over the next two sessions is how we're going to build on the basics of what Anna showed you, which was a great grounding in Galaxy to do larger and more complex workflows. And as I maybe promised or hinted at, I'm a huge fan of workflows in Galaxy. What Anna was saying near at the end there about you can reuse them on similar data sets is absolutely true. But by extension, you can tune and build your workflow on a subset of your much larger data, get it right, see that the tools are operating, producing the styles of results you need, and then execute those workflows on your much larger data sets or your collections of files, which could be tens to hundreds of files or more. So workflows really are at the core of your job and your analysis much more efficient. So I've got a couple of summary slides and then we'll have a quick question. The upcoming training sessions that are shared previously in the slides. Yes, to read this, Galaxy is supported by over 8,000 tools. So one of the things we're always asking our users to do is if you can't find a tool on a particular Galaxy service, get in contact with the operators of that service, explore the Galaxy tool shed and see if you can find a tool that can do what you want and get it installed on that Galaxy service. In terms of the actual platform, well, it's been in over now. We don't see it going away. It's well funded and well supported. There's resources in building flows, and you're going to see a lot more about what we really multiple ways of bringing data in, and importantly, you can also bring in quite complex metadata. So if you want to be tracking your samples by sex, exposed, unexposed, geography, age, or any other metric you can think of that is possible inside Galaxy. And we talked about the reference genome. So yes, almost in closing, we will be focusing on what Galaxy can do for SARS-CoV-2 monitoring. Here's a reminder on the bottom left for that Nature Biotech correspondence that covered it, and just a bit of an infographic showing how that in this particular paper, we can ingest Oxford nanopore, long-read sequence data, Illumina data of a variety of sorts, be it amplicon or RNA-seq. You're generating the kind of files you need, which are variant call files, all put through into a reporting template and then pushed out to dashboards or to places that can use to inform public health. Oh, sorry, I almost skipped over the team. In terms of Galaxy Australia, I showed the large community photo of some, but certainly not all of the global Galaxy practitioners. Well, here is the cast that keeps Galaxy Australia up, running live and updated with all the latest tools. And down the bottom there, are all our funding partners. And again, this just shows how much of a complex, but also a robust platform it is when so many organisations in Australia are willing to put computational resources, money, or FTE into keeping running. So with that, please feel free to email me if you do have questions. Idris has responded, thank you for where we are for session 2 and 3. We'll be closing this one shortly, coming back for session 2, where again, I'll give a slight intro, hand over to Anna to talk about genomics, come back to me for some chemoinformatics and proteomics, and we'll take a pause for the later session in the day where we'll dive much deeper into the complexity of the COVID genome and the variant calling. With that, we're at the end. Thank you very much for your attention this morning.
Thank you very much, Gareth, and thank you, Anna. That was very, very interesting start, and I'm really happy to see that there are quite a lot of people who turn up, and thank you so much for participating in this session. And there wasn't a question about where this location is going to be. It's exactly in the same spot. So no need to go anywhere. Come back here at 11 a.m. UTC +6 time, Bangladesh time. So with that, you can also, in the meantime, send any questions to Gareth or Anna. Emails are on the web page. So thank you again, everyone. I really appreciate that you've set aside time for us, and we'll see you at the next session. Bye for now.
Platform and the analysis of SARS-CoV-2 data and variants. The first, well, let me just launch in. We had quite a few attendees in the first session. I'm expecting a number of these to come back here. So we have some background slides, but I'll start on those, and as people join, that's fine. So reminder, myself, I'm Gareth Price. I work here in Queensland, in Brisbane, in Australia, for the Queensland Cyber Infrastructure Foundation. But today I'm wearing one of my hats, and that is the hat as Service Manager of Galaxy Australia. I'm joined by Anna Symes, one of our lead trainers and Galaxy experts, and she's based at the University of Melbourne. Idris cannot join us for this session and sends his apologies. He'll be joining us for the third session. And this particular session is to cover off some elements of analysis of SARS-CoV-2, in particular the genomics, chemoinformatics, and proteomics. And we're going to delve into them at various depths of detail based on some different activity. For those of you that joined the first session, there will be a little bit of a recap, but not too much. So really, the question posed here on this slide is, as we all understood, we were entering a global pandemic, and every country's response to that was slightly different. The scientific community's response was really to gather data, and we saw some fantastic early efforts in sequencing the SARS-CoV-2. virus. We saw proteomics efforts in visualizing critical proteins and its capacity to interact with our cells. And the Galaxy project, what they saw in particular was quite a bit of variety in the analysis methodologies which were being utilized by the community to analyze SARS-CoV-2. And in some, and thankfully rare cases, some just outright wrong styles of bioinformatics were being performed. And so the Galaxy community got together very rapidly to deploy a series of globally consistent pipelines, or in the Galaxy language, workflows, to enable a consistent approach to an analysis of SARS-CoV-2. In particular, for us in Australia, this interaction started in particular on a Friday afternoon, where we received a call from our overseas colleagues to say, would you be willing to be involved in global SARS-CoV-2 analysis? And we very enthusiastically said yes. At the same time, the two supercomputing centres in Australia, NCI, in our national capital in Canberra, and Pawsey in the state of Western Australia, both were running COVID-19 accelerated access initiatives. And this was designed to give researchers that were analysing SARS-CoV-2 access to the best high performance compute and cloud compute available in Australia. So we applied for that initiative as well. We were given an allocation and very quickly enabled these global Galaxy workflows on our supercompute centres. And that resulted in this Plus Pathogens publication, which was describing that an initial flurry of activity to deploy COVID analysis globally. It very quickly evolved into a mature product, and that mature product had 6 different headings. So genomics, and that will be a focus of this particular session here. Evolution, so tracking of the variants in observable samples carrying, or people carrying SARS-CoV-2. Chemoinformatics for screening of potential drug targets for the main protease of the virus. Proteomics for a number of different aspects, both and quite fascinating ones, and we'll get to those later, around growing the virus, describing its interaction with other microorganisms and the interaction with the human body. The Arctic analysis, which is amplicon-based data analysis, and another fascinating one, which is direct RNA sequencing to allow you to describe the viral structure. So this is what was deployed and rapidly developed globally. It all ended up here on a website that actually, Anna, if I can put you on the spot, if you could drop that web link into the Zoom chat would be great, thank you, which is covid19@galaxyproject.org. So this is a repository for all that activity that is currently going on in the SARS-CoV-2 space. If I go to that website right now, here we are. Here are all those topics as described across the top here. Down here, there is a live dashboard of SARS-CoV-2 monitoring, so you can look at the number of samples deposited and the number of samples analysed. It's by country and by time. So it's quite a lot of data. It just takes a couple of seconds to load. And when that does, you'll see you can select by region or country, and then by time for the number of samples. So here it comes now. So for some reason, and I suspect it's because where Sergey is based, the default country is Estonia. We have no problem with that, but you can see time periods for different countries. And the most comprehensive here is COG in the UK, and graphing their data over, in this case, the period of September 2020 to February 2021. From memory, this pulls in about 68,000 pieces of data. So again, it just takes a few seconds for this to spin up to show the whole picture. Now, I'm aware that I'm just getting a Zoom warning about low resources, so I'm just going to disable my camera for a moment, just to make sure. And A request to the attendees on this call, if you could keep your microphone muted unless you have a question, that would be great. Please feel free to ask your questions and to raise your hand if you want to ask anything in particular. So no apologies, this time period is nearly 30,000 samples analysed. You can see it tracking with, I imagine, what were the outbreaks in the UK, the particular number of variants observed, the variant density and the style of variant, and the most common variants found in that data set. And all of this has been enabled through Galaxy and through this particular site. Just give me a minute. Just going to, apologies, I can't. That's okay. I thought I could mute people. We, like I said, are basing much of the rest of this session and the next session on the information you'll find at this website. So these include pre-processing, assembly, and evolution pipelines for COVID, variant analysis, and phylogenetics, just to highlight a few. And what's really important about the way that the Galaxy community has approached COVID is that we have made these pipelines, I'll use our language, made these workflows, these Galaxy workflows, available on the Galaxy service. So we're looking at a screenshot here of Galaxy Australia into the Zoom chat to discover this website. These are just an example of some of the pre-computed, pre-available workflows that you have to access on our service. We went and had a look at the dashboard. So here's one of those periods. In fact, this is now the same period, but 1 year shifted, where again, you can see the rises and troughs in sequence, many tens of thousands of samples that have been not only Galaxy and been made available. We have talked a little bit about Galaxy Project, but I recognise that not everyone on this particular call may have been on the previous call. So I just very quickly want to show you what usegalaxy.org.au looks like. So this is our home website, home for Anna and myself. This is the Australian service, and give you a very quick orientation on how the service works. This is in no means comprehensive, just to give you a highlight for where we're going to go for the rest of the session. So from a geography point of view, the website contains on the left-hand side many, many hundreds of tools. In our particular case, it's about 1,500 tools covering various topics for genomic manipulation. Further down here, whole toolkits for styles of analysis like single cell and RAD-seq, and further on to climate analysis and proteomics, etc. The right-hand side of the screen contains your data, and this is explorable from oldest data sets at the bottom to the most recently generated data at the top. All of these are viewable by clicking on particular icons, editable, etc. I won't show too much of this because I'm probably spoiling the surprises Anna will give for you. It is a website, so this has certain advantages. One of those advantages is once you've uploaded your data, and you can do that by your local computer, connecting to a remote computer, or with a public URL, so a URL that doesn't require a password or a user. Bringing your data into the service, you're actually bringing it to our central repository. Anything you do after that is simply interfacing with a website. You can shut this down, you can turn your, all you're doing is checking there anymore. So you're not bound by your own computer's resources, you're not bound by the login period you have. This gives us a lot of flexibility to do work. Third and final, just very high level feature I wanted to show, and it's really any of these here. I'm going to go for quite an ambitious one. These are workflows. Workflows are the, pardoning my pun, the workhorse of the Galaxy system. These are a way to take many tools, and in this case, each of these blue boxes represents a single tool, build and chain the passing of data from one tool to the next. So this particular data set is used in 3 different locations, this one in 1. The results of this particular data combined with data from up here and all pushed to a third tool over here. It's a very rich and complex workflow that you can manage, you can audit, you can share with your colleagues to allow them to run it or check it out. Workflows mean that you don't have to sit here executing each of these tools one at a time. You do it en masse. And with that, I'm going to unshare my screen and pass over to Anna, who is going to take you through some of the genomics work that is presented in the COVID site, and then she'll return to me later to cover chemoinformatics and proteomics. Thank you, Anna.
Thanks, Gareth. I'm just trying to share the correct screen. Can everyone see that? I have Galaxy open in one window and a tutorial open in a second window.
Thank you, I can, Anna. Yep.
Great. So yes, thanks, Gareth. Today we're going to look at analysing some SARS CoV-2 genome data using the Galaxy platform. So some of you would have been in the session this morning where we went over the brief introduction to Galaxy. Some of you would have used Galaxy in other places perhaps. Either way, if you'd like to try and follow along this workflow today, you're welcome to. You're also welcome just to watch the steps being demonstrated, and everything is captured in the published tutorial, which I'll just paste into the chat. And that's something you can look at as we go or afterwards as well. So to get started, if you're following along today, I recommend opening the Galaxy Australia link. So that's usegalaxy.org.au. And if you've used Galaxy before, you can go to the top panel, user, and you can log in. If you haven't used Galaxy before, you can register and log in. Although having said that, if you haven't used Galaxy before, this may be quite complex to follow along with as a first tutorial. So yes, it might be better to start with some of the more introductory tutorials if you need to get familiar with Galaxy. But nevertheless, I hope the content in today's workshop will give you a view of what's possible in Galaxy for SARS-CoV-2 data. So I have the Galaxy window and I have the tutorial window. This is a tutorial which is in the Galaxy Training Network. It's by Wolfgang Meyer, and it's about assembling the SARS-CoV-2 genome. So I'm going to go through the tutorial and show the main steps, but to run in real time, this would take longer than the time we have today. So I'm going to mainly be using a history that I've already run, and I'll show you the workflow, the tools, and the outputs for the main steps, and then you can run it in your own time. So for this analysis, we have samples from people with COVID. In this tutorial, we'll get the data that we need, we'll get it into Galaxy, we'll trim and filter the poor quality reads. From those reads, we're going to remove human DNA so that we're left with just the viral DNA. And then we'll assemble those sequencing reads to hopefully generate the SARS-CoV-2 genome. So let's start with importing the data. In the tutorial, there is a full description of where this data is from and advice for the different ways you can get it into Galaxy. To save time today, you can import the data set that we're going to use from a shared history. So in Galaxy Australia, you can do that by going along the top bar, going to shared data, and then in their histories. And this history is called Published Completed History for SARS-CoV-2 Genome Assembly. So there's a few different histories in there you might see. The one we want is the genome assembly one, not the variant calling one. So if we go to that history, in the top right, you will probably see a cross button that lets you import that history. Because I'm the owner of this history, my button just says switch to this history. So I will do that now, and if you'd like to, you can import that into your own Galaxy window. So in this tutorial, there is a workflow that is linked here in the published tutorial. I've updated a few of the tools so that it runs well on Galaxy Australia today, and I've resaved that here within Galaxy Australia. So we can get that workflow by going again to shared data in the top, click on the drop down arrow, and then click on workflows this time, not histories. So workflows. Then the workflow we're looking for is the SARS-CoV-2 genome assembly workflow. To get that workflow, we'll click on the drop down arrow and you should see an import button. So click import and then bring that into your own Galaxy account. So the next step, and I should say, if you're having any issues, please just ask them in the chat window for Zoom and we'll see if we can help you. Otherwise, there's lots of extra help information in this tutorial. I know I have to go a little bit fast because of the time we have today, but I'm hoping to give a good overview. So now we want to view the workflow. To do that, we go across the top again in Galaxy and we go to workflow this time. So because you've imported the workflow, it'll be in your own list of workflows. So now we go down and look for it. It's the SARS-CoV-2 genome assembly workflow. And let's have a look at how this looks. So click on the drop down arrow and we'll click on edit. And that will bring us up what we call the workflow canvas, which is, as Gareth showed in his slides, a place where we can see all of the tools and how they join together. So at first look, this looks a bit squashed and we can't really see what's happening. So I will firstly just collapse the two side panels in Galaxy with some little arrows to give us some more room. I think I'll just make a little bit more room over here. And then I'm going to zoom out just so we can get an overview of what's going on. So this is a good place where you can zoom in, you can zoom out, you can see the inputs. Here we have short read data and long read data in this particular workflow. You can see how they connect to all of the different tools. And then you can see the outputs. So if we follow it through, not in detail, but just as a sort of a distant view, we can see the workflow, lots of different things happen, and we end up with an assembly tool and with a SARS-CoV-2 genome assembly. So looking at it in a bit more detail, for example, we just go back to the start of this workflow. Here we have our short read data in this box here. The first place that goes is to a tool called FastP, which is a trimming and filtering tool. If we click on that FastP box and just open up this side panel again, we can see lots of information. We can see the version of the tool in the top here, and then we can see all of the settings that are on by default for the tool in this workflow. So for example, here in the filtering step, if we click on filter options, we can see there are certain things that have been set here. These values here, 20, 20, the length filtering, we want a minimum length of 50. There's a lot of detail here in the default tool settings that have been set up with the workflow. And you can change those here in the tool panel, or you can also change them when you run the workflow. But if you want to see what's happening in a workflow, I think it's always good to look at the canvas of the workflow steps. And then if you want the detail of the settings, because that's often quite important, you can look within each tool for the settings that are on by default. So now we'll press the home button to go back to our main screen. So I will create a new history with the plus button up here. And let's give it a name. Call it SARS to genome assembly. So what we want to do here is we want to run the workflow to run all of those steps and result in this assembled genome. So the way that we do that, could everyone just make sure they're muted? Thank you. And any questions, please ask in the chat. So let's run the workflow. So we'll go to, again, the workflow button in the top here of Galaxy. We'll find this workflow again that we want to use, which is the SARS-CoV-2 genome assembly. Then we just drag this bar over so we can go all the way to the right there. And the run button is over here. Click on that. That now gives us lots of options here in the center panel. So we are going to change some things in here. Let's just check everything's correct. Oh, that's the wrong workflow, sorry. I've done what I said not to do. Let's just find the correct one. Okay, SARS genome assembly. Make sure I've got the right one. That looks better. Right, we need to check the inputs. So the collection, oh, I should go back a step. I should be in the history where I imported the data. So I'll just go back a step and make sure I'm in the correct history. So that we got that history from the shared data, histories. And we're looking for the one that is the base files for SARS-CoV-2 assembly. That's here. I'm actually going to make sure I make a copy of that so I don't alter the one that you all are working with. So let's just make a copy of that for running it today. To do that, I'm clicking on the button to view all of my histories. And I will actually make a copy of that history. And then I can run it from there. Go back to the home button, back to the workflow button and the assembly. Make sure I click on the right one. Okay, so let's check we're giving the workflow the right information. We want to use the paired end data with the Illumina tag. Then we wanted to use the correct long read data. And that's probably all we need to check when we are running this workflow. Just check we've given it the right inputs. And then we can click Run Workflow. So if you're following along today, you can start that workflow running, and it may take a few hours to run, so you won't get the results back probably within the time of this tutorial. But what we can do is we can look at a full published history of the tools that have already run, and we can have a look at the outputs. So to do that, we can go to Shared Data, Histories, and we have a completed history here that we can have a look at. So you can import that history. And I'll take you through what some of the outputs are so that in your workflow that you're running now, later on you can check that you've got the same outputs and that everything worked okay. So this is a big list of output files from that workflow that we've just run, from the top all the way to the bottom. There are some hidden files in here. If there's something you can't see, you can always opt to show the hidden files or not, but sometimes they just clog up the view a bit, so you might prefer to keep them hidden. We'll look at some of the files now. If we go right down to the bottom, we've got our input files here. These are our input reads. We've got Illumina reads and Nanopore reads. We can have a look at them if we want to. These are we've got 3 sets of Illumina reads. They're all paired end, and click on the eye icon and we can actually see the contents of that file. So this is a FASTQ read file. And we also have our Nanopore data as well, 3 samples as well. Another FASTQ set of reads. So what we did in this workflow, first of all, for the short reads, we got some information about their quality. And when you're not familiar with a workflow, you'll probably be going back and forth a bit to look at it in the canvas to see what tools ran where. So let's do that again here, just to double check what we're looking at. In the workflow list, let's just remind ourselves of that first step. So we had our short reads. And what is happening in this workflow is that the short reads are going to the FastPeak tool. And after that, one of the outputs from there is actually going to another tool called MultiQC, which gives a nice visualization of that output. Let's go back to our current history. So let's look at the MultiQC output as one of those outputs in the whole workflow. So here that is item number 52 in our history. We can click on the eye icon. And this gives us a visualization of the results. So there's lots of different information in here that might be relevant depending on the analysis that you're doing. One of the things we'll just look at here is that if we look at the filtered reads section, we can see we've got our 3 samples here. And from this, we can see 2 of our samples are quite small and 1 is much larger. The larger one seems to have a lot of quite low quality reads in it though. So all that sort of information is going to be useful when you're doing probably most analyses, and you'll probably want to look at a lot of the information within the QC reports. Okay, so the next step in the workflow, I'll just go back to that. So I think it's useful to sort of refresh what it actually looks like in terms of the steps. So after those reads went to FastP for trimming and filtering, then they were sent to a tool called Bowtie 2. And within this tool, they were aligned to the human genome. So we can see that by clicking on Bowtie here in the workflow canvas, and we can scroll down, look at the settings that were the default settings, and here we can see the human, oh no, the selected genome is the human genome here. So let's go back to our results. So the result from the mapping to with using the tool Bowtie is file 53. We'll click on that and we can see there are 3 outputs. Each of these outputs is a BAM file. So this gives information about where the reads map to the reference genome, which in this case is the human genome. So they don't really mean much by looking at them like this, but if you just want to refresh your memory on what a BAM file is and what information it contains, you can double check all of the outputs like that. And then we also got some information on the mapping statistics, and that is file 54. We can click on that, and again we have 3 output files. We can click on the eye icon for each of them and get a lot of information about how well the mapping worked. I won't go over the detail of the outputs here, but just to show you what the outputs are in general from this workflow. So in our workflow then, the reads that didn't map to the human genome, they're the reads that we're interested in, because these are the reads that contain the viral sequences. So in our workflow, these were converted from the BAM format back to FASTQ format. And then in this particular workflow, that set of reads was very large, so it was subsampled, just so that the assembly in this case would be quicker to run, and it didn't change the results much because there were so many reads in this case. So after all of those steps, after the subsampling of these reads, we got smaller sets of reads. In this case, it is files 85 and 86. And so these are our forward and reverse Illumina reads that are ready to go into the assembly tool. So another part of this workflow is the long reads, the nanopore reads. So with those, we ran nanoplot on those to get some information about their quality. So we can click on those results there and delve into them further, but I won't do that here. But the nanoplot reports are quite nice, lots of information about the reads. And then as we did with the short reads, we matched the nanopore reads to the human genome, extracted the non-mapping reads, got them into FASTQ format, and then they were also ready for analysis with the assembly tool. In this case, in the workflow, the assembly tool is called Unicycler, and we can look at the output there. It's file number 88. Let's have a look with the eye icon. So these are the assembled contigs in FASTA format, which is not that useful just to look at in terms of a FASTA file. So there's a really nice tool available called Bandage, which shows you your assembly. And we can look at that here with file number 90. on the eye icon. I'm just going to collapse the sides again so we can see this a bit better. So this is really nice. This is the assembly here. What we can see is that the longest contig that was assembled is contig number 1. It looks like it's almost 30,000 base pairs long, which is what we would expect for the SARS-CoV-2 genome. So that's really nice to see. And there's also lots of smaller fragments that were assembled here. And the tutorial has lots of information about how you can explore all of these contigs, but perhaps in particular the smaller contigs to see what they are. They're likely to be from contamination because we only removed the human DNA in this case, not any other potential contaminants. So there's information there about how you can work out what those other contigs might be. So in summary then, so we ran the workflow. Again, there's lots of detail in the published tutorial about that. If you're running this the first time, you might want to do it step by step so that you could see all of the steps, how each tool worked, whether you wanted to vary any of the parameters in the tools. In our case here, by running the workflow, we have all of the outputs of the workflow here in the history. The main one that we're after today is this assembly of the genome, which we've got, which is nice. And then that is then ready for further analyses downstream, particularly for things such as genome annotation. Yeah, so that's the summary there. Gareth, did you want to add anything about that particular workflow or in general?
Sorry, I was not unmuted. Sorry, just to have a quick query I'm just addressing. Yeah, just give me 2 minutes to address that.
So I'm just going to paste in the link to the tutorial again into the chat in case anyone missed that. And as Gareth said earlier, this is one of many tutorials in the Galaxy Training Network that cover lots of different topics, but this is quite a nice one if we're looking at COVID data, which we are in today's sessions. And it's a nice workflow with lots of different steps in it. So if you're also getting into a more intermediate Galaxy level, it's a good way to start seeing the complexity of workflows and how they run.
My apologies, Anna. My apologies, everyone.
That's all right.
Dangers of work from home means that there are always a variable you can't control. Are you nearly finished, Anna? Because I might just follow this on with some data upload commentary, if that's the case.
Yeah, that's finished now. So, yep.
Excellent. And a reminder to everyone, this session was listed at 1.5 hours, potentially broken into 2. I think we may just push on with the next part of the demonstration because it's not going to take a full 45-minute session. If you get a little bit of time back in your day, I imagine most people aren't going to complain too much. So I'm just going to share my screen. What I just wanted to emphasize, because this is one of the elements of Galaxy that we have a lot of questions about, or conceptions, not misconceptions, just questions about how to operate the service. And these reside around how to get your data into the service. So I mentioned that once your data is uploaded, it's at a Galaxy service, and that's fantastic. That's where you want it to be. And you can do analysis. Now, Anna has come and shown you a fantastic, but you can imagine quite scalable piece of work. So assembling not only one SARS-CoV-2 genome, but assembling many from all the patients that you're viewing or critiquing in your particular population. And so what you want to be able to do is bring data in efficiently, bring it in en masse, and so therefore have it ready for your workflows. So I just want to highlight here a little bit of off-script activity, which is what really is contained in this rather non-threatening data upload menu. So working through the various options here on the screen, choose local files should hopefully be fairly obvious. These are files on your particular computer. They also have the capacity to mean that if you want to push a file from your local computer up to Galaxy, you do need to have an active internet connection the whole time. You do need to leave your computer on the whole time. None of that is bad, but it just makes management of uploading your files more complex. If your files can be exposed by a URL, so a hyperlink, this is a fantastic way for Galaxy to administer the uploading of data. So Galaxy in the background will talk to the server that that URL is pointing to and to Galaxy. You can actually shut down your Galaxy and that upload of data from the other server into Galaxy will continue. Now, many people don't necessarily want to expose their data via a public URL. URL organization that funds Galaxy Australia. So they pay for data sets of national importance to be generated. These include geological sampling, marine species, and many other data. And they have a similar situation. They want to get data into Galaxy. And what we have managed to do in URL. Apologies, it looks like I dropped out for a minute there. Hopefully not too long. Just coming back online now. Yeah, so I just want to emphasize that you can bring in URLs and you can bring them in a fairly secured fashion, depending on how you choose. There are two other aspects I just quickly want to highlight before I'm going to move on to what's the second half of this particular session. This is rule-based uploading, and it doesn't look particularly dramatic here. There's a fantastic tutorial to follow it. This allows you to bring in not only multiple URLs, but bring in multiple URLs where you have metadata associated with those URLs. And the final thing I just want to show before I move on is remote files. And remote files are a slightly new addition to the Galaxy suite of activity. And this is exposing repositories of data. So this particular top one here, Cloud Store, this is an Australian service for Australian researchers to store data, but it is analogous to Dropbox. It's analogous to Microsoft Azure or Amazon storage. It's a place for you to hold your data in a secure manner. Here are all some public data sets where you can bring in data, for example, here, the COVID-19 data lake, a central repository of up-to-date and curated data sets related to SARS CoV-2. So don't be worried if you're going to think about bringing in large pieces of data. Galaxy can handle it, and it can also handle the administrative aspects of downloading or uploading large data sets. So with that, I will get back on script. Let me just get my computer ready. And we're going to switch tack a bit and talk about some other aspects of analysing SARS-CoV-2. And these ones come with complexity. And I don't mean to suggest that the genomics is not complex. Genomics is where Galaxy started. It is where it's most rich in tools and support, but it has also benefited hugely from the core basis of data generation in genomics, which is essentially that we have 4 very, very predictable bases of DNA, that these can be stored in very predictable file formats, and that this gives the community of data generation in the genomic space and consistency. It doesn't matter if your data is from Illumina, Nanopore, BGI, Thermo Fisher, PacBio. It can all be expressed in a very common language associated with DNA bases. This is not necessarily true when it comes to the chemoinformatics and the proteomics spaces, where the variety of data generating mechanisms means there are a lot of different ways to have your data expressed. Nonetheless, we are about to have a look at one of these protocols. In particular, this comes under the chemoinformatics or computational chemistry heading. And this is a virtual screening of the SARS-CoV-2 main protease using a particular program called RDoc and some scoring via POSE. As I said in the first session, and as Anna has highlighted just now, these protocols are available on the Galaxy Training Network. They come as orientation with recommendations of what to do before this, if you haven't done them before. They come with all the necessary data sets and workflows and provide both a comprehensive introduction, background, etc. Stepping through the publicly available data, through the tools of execution written here, nice pop-up boxes if something changes between the highly cited reference this came from and the operation of the tutorial. And here again is the paper that's driven by this tutorial. So we will be stepping through this, and I have some excuses to make, and I apologize in advance for these excuses, but you'll see them as they come up. So a little bit of context first about what we're trying to perform. So we have the main protease. This is the compound in question. We can do particular things to it. First of all, we can model it so we know what space it takes up. We can, which is all here, sorry, in the protein preparation. We can then use the ligands or the molecules we've prepared previously to expose them to the active site on the protein, score these ligands for their capacity to interact with the main protease, which is the scoring down here, and then filter and rank those till we find the best virtual molecules available to us. And I think there's a couple of really important things in here that I would like to emphasize along the way. And they are statements like this, which look particularly benign, but are not, like split files into chunks. So one of the things we haven't really been able to show you on the workflows yet, but I'll show you shortly, is that each of those workflow steps, whilst it looks like a single workflow, those steps in themselves can be performed independently if they have no prior condition. So if they don't need a previous piece of information, Galaxy can schedule them in parallel. And this particular step up the top here, this ligand library was split into many, many small files to allow for a lot of parallel processing that sped up this process. You're going to see me do it with only a short few hundred number of ligands, but when this was processed on, I think, up here, the 64 million, obviously the ability to split this file was a fantastic way to speed up analysis. So what is all this based on? Well, again, it's based on this flurry of activity that occurred as we understood we were inside a global pandemic. In this particular case, It comes from the UK National Synchrotron Science Facility, which is the Diamond Light Source. They are built on previous work, as cited here by Professor Z. Rao, who had crystallized in a very short order the main protease of SARS-CoV-2. He made a fantastic gesture there, as all of the open science is, to make that available for other people to build on. The Diamond Light Source facility built on that activity by producing a high-resolution image of that main protease, and cited or noted that critically, this showed that the active site in the crystal structure was both empty, so ideal for modeling, and also solvent accessible, so could be a good site for modeling as well. That is available here, and Simon's history on Galaxy. I have it preloaded here just so you can see the molecule here, and also so you can see just the style of visualization one can perform in Galaxy. So this is a 3D protein structure that is visualized in Galaxy, and as operators of a national Galaxy service, we're seeing these as becoming increasingly more popular for our users to perform. So on the basis that the main protease molecule exists and that we have a training course, which is here, we're now going to go off and look at a number of different features to see how this was performed. We'll skip back out to the training protocol in a minute. And in this particular case, we're going to make use of a different Galaxy. In this case, it is Galaxy Belgium, before returning back to Galaxy Europe and finally having another look at the Galaxy Project website. Now, the reason we are, in maybe loose terms, shopping around for Galaxies is for this particular statement here, available on these Galaxies. And I highly recommend, one, that you come to the traininggalaxyproject.org website to look at what the content is, but also to check where this particular training event or training course can happen. And in this case, you'll see that there are a number of websites that support this particular training topic, but Galaxy Australia is not listed here. It would be down at the bottom. There's no particularly good or bad reason for that, other than that each of the Galaxies works with its own local community. And we haven't necessarily preferenced this tutorial yet, is probably the best way to put it, but it will turn up on Galaxy Australia. Now, what has happened, and hopefully I won't be made a liar in the next slide here, is Galaxy Europe. Here it is here. Yeah. So Galaxy Europe has been undergoing a maintenance and improvement cycle since last week, but in particular since we planned this event. And they are the main supporters of this particular tutorial. And they have been the drivers behind much of the COVID at galaxyproject.org activity. So unfortunately, at this point in time, I can't show you real computing against the chemoinformatics database, as Anna was able to do with the informatics, the genomics. So we're going to step through what some of the results would be. And as I said just before, we'll make use of some alternative Galaxies. In this particular case, we're going to make use of a publicly available Galaxy that is mostly supporting this protocol, which is Galaxy Belgium. So you will see, much like what Anna just demonstrated, there's a particular cadence or rhythm to following the Galaxy training network. And if you become familiar with this, you can start to upskill yourself in a huge number of different topics within the bioinformatics space. So we, again, strongly encourage you to jump onto the training@galaxyproject.org website to follow along with some of these materials. So I'm not going to particularly belabor the background. I've said much of this. The important things to talk about in here are that the initial list that was originally thrown at the main protease was 41,000 candidate molecules. However, in this particular tutorial, we're not going to analyze all 41,000. And that is because that original and blunt force approach took what we do as CPU time, 20 years of CPU time to consume, not counting the graphical processing unit time. So the tutorial here is a cut down version of that library containing only 100 molecules and only containing one resolved main protease fragment. So this particular protein does carry a number of variants that change its structure somewhat, and those are the variants we're going to describe and explore in the third workshop later this afternoon. However, so here, to make the tutorial run faster, we're only going to use one particular visualization or resolution of the main protease and only 100 molecules. So as Anna has shown before, we go off and get our data, and this is held at Zenodo. If you're not familiar with Zenodo, this is a public repository of open data, and it's an extremely useful place to deposit a file if you want someone else to be able to analyze or access that file. We ingest that information and we'll go and have a look at it, and we rename it to the three file types here. I am getting a message, apologies that my internet is breaking up a little, and I do apologize for that. I have it running at a bare minimum. It will break up as I change screens, so I'll try not to toggle too much. Once we bring these files in, as Anna showed previously, it's always a good idea to go and inspect them, and we'll do that very shortly. But to try and minimize where we're at with my Zoom bandwidth, what we are going to do is take this list of potential ligands, our candidates, and we're going to put a charge structure over the outside of them so we can see how they would potentially interact with our docking site. So first of all, we're going to enumerate the charges on those molecules. Then we're going to do some conversion to file formats. And if we can think about one particular thing in Galaxy, and that is that much of your bioinformatics journey and much of the time that bioinformaticians spend is actually converting one file type to another file type to make something readily available from tool 1 to tool 2. So you see that within this particular tutorial, there's nothing different. You're spending your time converting the candidates to a new file format so they're accessible to a new program. And the last part of this, and I'll skip to Galaxy in a moment, is splitting the file. And this is very attractive functionality and can't really be overemphasized. If you have a finite amount of computational resources, rather than potentially throw one large file at it, can you throw many small files? In this case, we started with a file of 100 compounds, and we've asked to split this into 10 files. So it will end up being 10 files of 10 compounds that can be computed in parallel rather than sequential, and that speeds the process up. So what does this look like in Galaxy? I just zoom in a little bit. Here are our three initial input files. Anna showed this as well. Always a good idea to have a bit of a look at your file. Doesn't always mean they'll be human readable, and doesn't always mean that they will be intelligible, but nonetheless, it's worth having a look. I can see in here the various descriptions of molecules. I don't necessarily understand them all, but they're in there, so I can see what the file type is. I can see the file type of the receptor from the protein database, as described by position and various measurable features, and the number of hits on this particular molecule as in a database format. So we take those, we enumerate those molecules and do the charge calculation, and we end up with a larger file, but now expressed more clearly in terms of charge state. We convert that to the SDF file, and here is this split list. Each of these files contains 10 sublines in them. So it'll be about 39 molecules each, since we had 381. So the last file presumably will be a little smaller and is by 1 molecule. Collections are a fantastic way of doing work in Galaxy where you have a lot of files. I will show this simply by finding a tool. I'm going to use the same tool that Anna showed earlier. Just to give you a quick orientation on Galaxy, you can bring in a single file, you can bring in multiple files, but this means you have to select files from a list, and this is manual and potentially error-prone. Or you can build a collection of your files, as we have done here, and this collection then is a single line entry, and all that data and metadata management is held into the collection. I am just pausing between each screen flick just in case this helps with bandwidth. So we've split our collection, and now the next thing to do is prepare the active site. So again, we are doing conversions and file conversions to prepare the data for something that can be accessible to the programs that we need. This can be a challenge in Galaxy. However, Galaxy as a software is particularly helpful to you. So if we go and look up compound conversion at a Galaxy service, and I'm going to use the Belgium one, given that that's where we know this tutorial works. Compound conversion. It is asking for one of these file types. So Galaxy will always prompt you for the file type, and in fact, in many cases, it will not accept a file type that is not from its selected list. So yes, you'll need to do file conversions, but this is actually a good thing because it allows you to manage your data journey carefully. So more conversion. Then we're doing something here, which I want to point out is a feature of the training portal, which is questions and answers. So if you're reading through this tutorial, often you'll be posed questions just to see that you're on track with your learning activities and a solution. In this particular case, we are talking about building a ligand from all possible sites within that active site that becomes our Frankenstein ligand, something that's a bit of everything and none of itself, but this is an idealized squish into that active protease site. We build that, then define our active site by a number of criteria. And again, as Anna showed previous in her first tutorial on the basics of Galaxy, one of the real powers of Galaxy is the ability to rerun a job and simply alter one feature. So this has been set up very carefully and designed to allow those molecules to bind. But if you were curious about any of these settings, you could rerun this tool again and incrementally change these values. Finally, we expose that prepared ligand list and the prepared active site together and perform the docking calculation. After we do the docking, then we have 10 files. We want to merge those files back into a single collection where we can score and rank the files. So if we come and look at our Galaxy history, we've prepared that Frankenstein molecule, we've prepared our active site, we have gone and scored this collection down here. So we've gone and scored all those 387 molecules, sorry, I was thinking 81, 387 molecules before we go and collapse that into a single collection of all the molecules, their charges, their positions that can fit into that active site. And that takes us, pardon me, that takes us most of the way through this tutorial. Unfortunately, it also takes us to the point of view, to the position where I can no longer show you this as a pre-computed tutorial, as these two particular tools on the screen here are only supported on Galaxy Europe. And as I said, they're doing some much worthwhile upgrading of their infrastructure at the moment, which has put their service as unavailable to us today. However, what I can do is show you what the results of that look like when you do a larger study. So, okay, hopefully we can get through this next bit without my computer bombing out on me. But I do apologize to everyone that, there's some circumstances out of my control. So in the final session, before we close, and we are running to time at this point, I wanted to also highlight one of the other features of the COVID workflow. Sorry. Hopefully you're now looking at my proteomics screen. And this is another one of the opportunities or workflows that's supported on the Galaxy Project website. This one is another one that unfortunately is mostly supported in Galaxy Europe, but it also is an interesting biological question. So it is more about showing you what you can do with Galaxy rather than the genomics, where really what we're providing for you is well-curated protocols and workflows for doing your analysis. So in this particular case, both proteomics and metaproteomics is supported on Galaxy services and is growing in breadth and depth. For those of you that are in the proteomics space, you'll understand that the data types generated are very, very different to genomics data sets. The software involved is often proprietary and black box. And there are moves now globally to change that practice. So Galaxy P, standing for Galaxy Proteomics, is a very large advocate for democratizing access to proteomics data, which is traditionally mass spec-based data, to allow some common and again standardized protocols to rise out of that activity. And we are starting to see that in software packages like MaxQuant. However, at the galaxyproject.org proteomics website, what we have are a series of test cases and explorative cases to show you what you can do with proteomics rather than provide you with a single pipeline like in the genomics space that really should fit much of your genomics analysis. This particular case, the mass spec data has all come from a global repository called Proteome Exchange that takes data from large proteomics producing facilities around the globe. And Galaxy Project didn't take on the primary analysis of this data, but rather reanalyzed the mass spec data sets to show you what you could do in Galaxy, and in particular what you could do without having to beg, borrow, or steal time on proprietary mass spec software. So the kinds of projects they did was the reanalysis of data from cell culture, from clinical samples, from protein-protein interaction, but also from a fascinating look at metaproteomics. So looking at the expression of proteins across multiple samples, and this was done in 3 clinical studies. The first particular example was a shotgun proteomics analysis of SARS-CoV-2 infected cells, and looking to see how you could optimize whole viral particle antigen production for vaccines. So how can we identify the best product material to induce an immune response to build antigens against, against a particular antigen. So the particular paper is here from 2020, growth of bacteria, protein precipitation in gel digest, followed by LCMS, and then data analysis looking at the styles of proteins that can be detected and doing pathway analysis for the host material. I think what I'll do shortly is put some of these links into the chat box, both here on Hoover, so that you can explore this yourself. These are all well cited at Galaxy Project, but I don't want to further undermine my capacity to share multiple pieces of data with you. So I do want to move on then to the second of the two studies, which is metaproteomics. And this was quite a fascinating look at 3 different studies. So gargle studies from the back of the throat, nasopharyngeal swabs from non-infected and infected patients, and upper respiratory tract samples from COVID-19 infected patients. All 3 of these were analysed in the one paper and all followed pretty much the same overall workflow that you can see on the right-hand side of the screen, which is to take those raw, publicly available data sets, and thank you again for the researchers in sharing that data, and putting it through Galaxy to identify peptides, putting those through a database searching function to remap them against PepQuery, and then to do speciation of those. And the speciation is quite fascinating, but before I get to that, the overall process, as I said, search the data sets, look for microbial peptides and determine the taxonomy associated with the peptides. That was using UniPep. Validate those by a secondary query search, and then look for the number of what are called valid peptides corresponding to microbial taxonomic units in much the same way that you would use 16S molecules to do operational taxonomic unit classification using ribosomal RNA via a particular tool called LoRaKey. So the workflow there on the screen is really just to emphasize, as with many of these workflows, there's a lot of tools that are working in synchronization, taking data from one tool, passing it through, at the same time manipulating other data to join it up before completing your final analysis. The paper in the end showed that it was able to detect a number of species, Streptococcus species and a Lactobacillus species, as well as, importantly, the SARS-CoV-2 in 2 of the 3 studies. So the first particular study didn't show any detectable peptides associated with those 3 species. In the second and third study, ND for not detected and the number for the number of detected peptides. Both of these species are common to the upper respiratory tract or the nasopharyngeal tract. And what's shown on the bottom then below is the confidence as expressed by all these metrics on the right here for the detection of those peptides and therefore the speciation of that individual in that study. So quite a very cool look at, can we detect multiple species by the peptides in a way that traditionally we probably would have done this via genomic detection. So with that, I apologize again for my bandwidth. Hopefully I'll have it secured by our 3rd session today. So I just want to thank the Galaxy team as shown here. Express that you feel free to ask me any questions here via Zoom or via the Hoover app. And in the 3rd session later today, we will be diving deeper into variant call analysis in Galaxy, as well as some of the advanced visualization features at the end of that particular session. Anna, and I believe, Idris, I saw you online if you have anything you would like to add. feel free. Otherwise, I think we'll be closing this session about 5 minutes early. Oh, thumbs up from Anna. Thank you, Anna. Okay, thank you, everyone. And yes, I will post the links into Hoover shortly.
Should I stop the recording?
Please.
Yeah, I'm stopping.
Okay, so welcome to the third and final session relating to Galaxy and its usage in the analysis of SARS-CoV-2 virus, both the data and the variants. So this is the third session. We'll be looking to explore and monitor COVID variants using Galaxy, and this is what we deem as the more advanced section of the training. It'll be predominantly run by Anna, who's done a fantastic job in the first two sessions already. I'm going to chime in at the start and the end, and Idris as well. So without much further ado, we're going to leap into this session. So again, I don't want to assume that people have attended the two previous sessions or have attended either of them. So just an extremely basic, almost too basic, but a recap of the platform we will be using. And that platform is Galaxy. In particular, today we'll be using Galaxy Australia, which can be found at usegalaxy.org.au. And I just want to highlight a couple of the very basic features just to orientate the more complex work that Anna will be showing. As I had some issues with bandwidth earlier to keep my video off for a moment, although I'm also not entirely sure why Galaxy is taking this long to load. Come on, Galaxy. There we go. So Galaxy in all of the global galaxies has the same look and feel. The left-hand side of the screen is a searchable tool menu key to particular activities or domains of research. The middle window is interactive between information, tool setup, and results. And the right-hand side of the screen is the data that you're going to be analysing or have analysed. So at the bottom of the history will be your raw data, progressing up through the analysis steps that you take on. You search for tools either by the tool name, and this is a conditional search, so I can type FAST for fast quality control, and this will filter the list shortly into all the tools that contain FAST in the title. You can see that obviously being a popular file format, there are a lot of those tools. So I could, for example, type FastQC to narrow down to the tool I'm looking for. And you can see there's only 4 of those. When I click on a tool, the middle tool menu, the middle panel changes. I can pick one, many, or a collection of many files as input. This actually is conditional, so I can only pick the things that conform to the necessary file input for this tool. I have the options for setting a number of different settings for the tool, and I hit execute. When you hit execute, the tool is scheduled onto that Galaxy service. So if it takes hours or in some cases days for that tool to run in terms of very big jobs, you don't need to leave this window open. You don't need to leave your computer open. You can just come back and check up on different times. You upload data into the service from your local computer, from remote cloud storage, such as commercial providers like Dropbox and Amazon and Google, or you paste in a publicly available URL, the probably easiest points. And the final point I make before I pass over to Anna to go through the last tutorial is workflows. If you haven't already heard about workflows, you're going to hear a lot more about workflows as they come through and through your Galaxy usage. A workflow is a chaining of tools together in an efficient way. And by efficient, I mean efficient for scheduling on the compute and efficient for you as a user. You only have to execute a workflow once to have a very complex series of tools run over and over again. In this particular case, there are many individual reference files all being combined together through a process of trimming, mapping, and quality control plus feature counts for an RNA-seq experiment. The user, in this case, would just need to run that workflow once to have all this data computed for them. That's about all I will say about Galaxy. So what we're going to do for this session before we come back nearer to the end, and I'm going to cover up some niceties at the end, is this particular tutorial here. And I'm not going to say anything about it. I'll leave it to Anna's very capable hands to cover this one off. Thank you, Anna.
Thanks, Gareth. I'll just share my screen and just double check. You can see Galaxy Australia and the tutorial.
We can, thank you.
Great. So yes, as Gareth said, we're going to look at analysing some SARS CoV-2 data with Galaxy. Earlier we went over an introduction to using Galaxy in the first session, and the second session we covered how to assemble those sequencing reads into a genome. If you haven't used Galaxy before, I highly recommend Googling the Galaxy Training Network. They have lots and lots of tutorials about getting started and doing more complex analyses on Galaxy. And if you have used Galaxy before, you can try and follow along today some of the workflows that we'll be demonstrating. So we're going to look at this quite excellent tutorial by Wolfgang Meyer and Berenice Batu. I pasted that into the chat window, and it's this tutorial here called Mutation Calling, Viral Genome Reconstruction and Lineage Clade Assignment from SARS-CoV-2 Sequencing Data. So we don't have lots of time today, so I won't be running the full tutorial, but I'll go over the main steps, and then at a later stage you can repeat the tutorial in full if you want to see every single step in it. So to get started, I have Galaxy Australia open in one window, and I have the tutorial open in another window. And what I'm going to do mainly today is to look at a history that I have pre-computed and look at the results from many of those tools. So for this analysis, we have samples from people with COVID. And in this tutorial, we're going to get the data we need and get that into Galaxy. We'll call variants. Then we'll see what genes those variants or the mutations occur in and what effects they may be having. And finally, we'll name the lineages that those different variants likely belong to. So to get started, if you've used Galaxy before, you can go to user and you can log in there. And if you haven't used it, you can register. And if you don't want to follow the steps as I go through, that's totally fine as well. You can just have a look at how it works. I've accidentally clicked on a link. And then it'll probably be clearer to follow it more slowly in your own time, step by step. But hopefully this will give you a good overview of the capability of Galaxy for analysing this type of data. And again, we're using Galaxy Australia, but there are various Galaxy servers around the world. They're free to use, and we definitely encourage people to use it. It's a great platform, really good for analysing small sets of data, big sets of data, lots of tools. So yes, we welcome people using Galaxy. So let's import the data that we need for this tutorial. First of all, in the top panel of Galaxy, go to Shared Data and then Histories. And in there, we will search for this history that I've pre-computed. So if we, in the search box, if we search for SARS CoV 2 variant calling, I'm having slow internet as well. There we go. We can see 2 histories have turned up there. The one we want is the one for variant calling. So this top one here, if you click on that and go to the top right plus button, that should let you import that history into your own Galaxy account. So you can do that if you want to. I'm actually already in that history, so I'll just stay in there. Okay, and so this history contains the input data that we need, but it also contains all of the results that we're going to look at. If you want to learn more about where that data is from and how you would get that into Galaxy or get other sets of data into Galaxy, please look at the tutorial that describes that in a lot of detail. It explains what the data is, where it's from, how you can organise it, and so on. But in the interests of time, I am starting with us having history with the data in it. So let's have a look at our data. You scroll down to the very bottom of this current history, and we can see our input files. And for example, our sequencing reads here, file number 41. If we click on that, we see all of our samples here. We can click on one of those, and then we can click on the eye icon. And that will bring up in the center panel that file displayed. So we can see this is a FASTQ file, and it has lots of sequencing reads in that. So back we go to the history. And we have some other files that we'll need for this analysis. So one of the things we have is a reference genome for SARS-CoV-2, and that is file number 2. So that's just the FASTA sequence of the reference genome. We have a table that converts some of the common names for gene products. That helps us in translating some of our output. That's the feature mapping file. And the data we're using in this tutorial, it's being sequenced with the Arctic protocol, which some of you or many of you may be aware of. And that includes a certain set of PCR primers. So because we're using that type of data, we've got an additional 2 files in here. We have a table of the primers, which is the amplicon info. And we have a bed file, which is a file showing where these primers bind to the genome. I'm just going to make this a little bit wider. So that's our input files. We have these 4 auxiliary files and lots of sequencing reads. So today we're going to run 3 different workflows. And so this is quite fast paced, and particularly if you haven't used Galaxy before, this may seem like a bit of a whirlwind, but hopefully, yeah, it does show a nice summary of the types of analyses that you can run. So to get the workflows that we need, we'll go to the top panel again, we'll go to shared data, and this time we'll go to workflows. First of all, we're going to search for we're going to search for Arctic PE data, paired end data. And that's brought up a few different workflows. The one we want is the one that's tagged with Galaxy or owned by Galaxy Australia. So if we click on the drop down arrow there, and then we can import that one. I won't do that now because I already have imported that. That's the first one we need. The second one we need is one for doing some reporting. So if we search for variation analysis reporting. Again, there are two that have shown up here. We want the Galaxy Australia one, drop down arrow and import. And finally, we want one for creating consensus sequences. So we're going to search for consensus construction. And there's the one we need there. I might just pause for a minute and just paste the links to each of those workflows into the chat, just in case anyone missed the names of them. So here's the first one. And I'm pasting into the Zoom chat, not the Hoover chat. So let us know if you have any problems seeing that.
I can move it over, Anna, if you need me to.
No worries. That's the second one there. And then this is the third one. These are all detailed in the tutorial as well, but these specific ones on Galaxy Australia are going to be useful for us today. So I'm just pasting them there in case you're following along today. Okay, so then we have imported our workflows. Let's have a look at them. Let's go to the top tab here that says workflow. And then you can see your list of workflows in your Galaxy account. And then in this workshop, we'll look at these one by one and we'll run them one by one. So we won't look at them yet in total. So the first one we will look at is the variant calling one. Let's see if we can find that in our list. It's the variation analysis workflow. We'll click on this drop down arrow and click edit. And this lets us view what we call a workflow canvas. So some of you will be familiar with this. I'm going to collapse the side panels here so we can see this more clearly, and I'm going to zoom out. So sometimes this can be a bit overwhelming when you look at a workflow to start with, because they can be quite large, but you can look at them piece by piece and really understand what's happening at every step and all of the settings. So another thing you can do is drag it around to an arrangement that suits you, which I always find really useful when I'm first looking at a new workflow. I like to follow through what's happening with all of the inputs and outputs through the whole workflow to really understand it. And because you've imported this workflow, you can actually make changes that suit you and then save it in your own Galaxy system. So what is this workflow showing us? We won't go through all of it, but we'll look at some of the main features. This box here, these are our input sequencing reads. And these are going to go first to the tool called FastP for some filtering and trimming. The processed reads from there will then go into a tool called BWA-MEM. And that's where they'll get mapped and so on. So when you have more time, you can follow all of the steps in the workflow and see what's happening to all of the files. And these are all discussed in more detail in the published tutorial as well. So we can see there are a lot of processing steps in here. There are some variant calling steps obviously as well, which is the main point of the workflow. And we can follow right through to the very end. Zoom in again. My internet's being a bit slow too. Well, if it doesn't show up, that's all right. But the very last tool in here is a tool that characterizes the effect of the variants that we've identified. So hopefully you're having better luck viewing that on your own systems with perhaps better internet. So other things that we can see in here, I'm scrolling back over to the start of this workflow. We can see this other input file, the reference genome. We can see how that's being used as input to various tools in this workflow. We can see another of our input files, the bed file, also being used as input to some of the tools. So it's a really nice way to visualize what's going on in your analysis and the tools that will be used. And a really useful thing in workflows, which I spoke a little bit about in the previous workshop, was the ability to see all of the settings for the tools that are being used. So you can click on a tool. I'll find a good one. Let's look at map with BWMM. And in the right-hand tool panel, you can see all sorts of things like the tool version that's being used and all of the default settings for this tool in this workflow. And if you want to, you can change any settings in here in the workflow to reset the defaults, or you can change some of these settings when you run the workflow. Okay, so that's our workflow that we're looking at. Let's go back to the home button. Okay, so to run this workflow, we would find it in our workflow list. So this is the variation analysis on Arctic PE data. So that's paired end data. And we would click the, let's check I'm running the right workflow here. Okay, this second one. So we would click the run button. That brings up the information in the center panel about the workflow. And we need to check we're giving it the correct inputs and change any parameters if we need to. Okay, so this workflow needs a paired collection of FASTQ reads. We will give it that. In our case, this is file 41. It needs the FASTA sequence of the reference genome. So we'll give it that, which is file number 2 in this case. It needs the bed file of the primers, and it needs some information about the primers, which is file number 3. So you want to check that all your inputs are correct here. Galaxy will try to guess what inputs you want, but it won't always do that correctly. So check the inputs are correct. We don't have any other settings we need to change at this point. Here we can see all of the tools that are going to run. This is a quite complex workflow. Lots of things are going to happen, and we don't need to change any of the settings here. So I won't run this workflow now because I have already run it. But if you were doing this yourself and you wanted to run it, at this point you click Run Workflow. And so let's assume we have done that and it's run. And we can now look at some of the outputs. So from this first workflow, there's 2 main outputs that we'll have a look at. The first one is the variant calls from each of those samples. So let's look at file 618. So each of these are the variants that have been identified in each of these samples. We can click the eye icon to view that file. These are all VCF files. And they're not going to be super informative just to read one by one, but if you'd like to see exactly what's there and what the tool has done, it's always good to have a quick check of your outputs. So each of these samples has its own file in VCF format. And another of the outputs we'll look at is the effects, the hypothesized effects of some of these variants. So this was a tool called SNPF, and this is file 638. So again, this has created an output file for each one of our input samples. We can click on this to have a look. I'll just compress the side panels. And this gives a lot of information, and I won't go over all of this information now, but it's really nice to have a look at in detail. So each one of these samples has this huge amount of information that's been computed about the particular variants, what type they are, and so on. So that's the main output from that first workflow that we've run. So let's run our second workflow now. We'll go to the top panel to workflow, and we'll find the workflow we need, which is this is the reporting workflow, which summarizes the information across all of those samples from the first set of results. Let's have a look at this one again on the workflow canvas. And again, I'll just make it so we can see this a little bit better. Zoom out. And again, you can see this is another fairly complicated workflow in terms of its size, but once you can see it step by step, it makes sense. So this workflow is all about just summarizing that information from the first set of results. And this input here, this variation data to report, that is an output from our first workflow. And then that goes into various tools, various processing, formatting, and so on. And we end up with, right across the other side, again, it's not displaying super clearly here, but there are three main outputs, and we'll have a look at them now in our history. So let's go and press our home button. Okay, so let's run this on that variant information from the first workflow. So Galaxy Australia, go to our workflow list, our variation analysis reporting workflow, that's the one we want. And we need to click the run button. So there's a couple of things we're going to change in this workflow in these settings. The first thing I want to do is I want to click expand to full workflow form, because there's a few things I know I want to change in here. So I'll click that, that brings up the full workflow here. So the things I'll change here, the number of clusters, so that's going to be in this case 3. The gene products translations, this is file number 1, which was just the translation of some of those terms into more sort of commonly used terms, a bit easier to read the output. The variation data to report, that is one of our outputs from the first workflow. In this case, it's file 637. That's our annotated variants. And one other thing we need to change in here is just one of the outputs. We scroll all the way down, again, seeing a huge range of steps that are involved in this workflow. To the second last tool, which is a plot, a variant frequency plot, and then image properties, plot format. We just actually want to change that to a PDF to display a bit better here. So then if you were running that at this point, there's nothing else we need to change. We'd be clicking run workflow. But because we have already done that in this case, let's now go and look at our results. So there are three main outputs from this workflow. The first one is, I should say, you might notice in the history there seem to be some numbers missing, and that's because there are a lot of outputs that have been set to be hidden because they probably won't be directly used and we don't want to clog up our view. So if you do find you're looking for a file that you think should be there, you can go to the top of the history and toggle the hidden button to get it to display all of the hidden files. We don't need to do that here. We just need to look at file 810. Again, clicking on the eye icon. So this is giving us the variant calls for the samples. Each line here is a sample. So it's summarized all of our samples into one output. And a full description of what all of these columns are is in the published tutorial. And then file 811, here we have one line per variant. So again, it summarized all of the variants of all of the samples. But probably the most interesting output from this workflow is a nice plot. And this is a plot, which in this case is file 1590. Before I click on the eye icon to display that, I'm actually going to click on this little grid here in the top menu, which just gives me a bit of extra flexibility in displaying the outputs. So now I'll click on that output. It brings it up in its own window. And this is a bit small, obviously. So let's just zoom in a little bit here. But this is a really nice plot. So again, there's a bigger description of this in the tutorial, but what this is showing is our samples have been clustered here along the left-hand side. Each row is a sample. The very top, very top line here are the genes, different genes, and the types of mutations are coloured in different colours under that. So it's a nice way to see how the samples are clustering and what sort of mutations are happening in what genes. If I zoom in even more, we'll be able to see the gene names better. So I think that's quite a nice plot to give you an overview of what's happening in your samples. So close that. Okay, so that's workflow 2. That's our summary of our variant calling and then putting it into report format. And something we're not showing today, but something that is possible to do is to take some of those outputs from the reporting workflow and to do further analyses on those outputs in other platforms such as Jupyter Notebooks or the observable dashboard. So that's something to keep in mind, that there are lots of other explorations you can do on that data, and that's talked about a little bit in the published tutorial as well. But we won't go over that today. And now finally, we're going to run a workflow to create consensus genome sequences for our samples. So let's go to workflow. Look for the workflow that is the consensus construction, and we'll click edit to again view that on the canvas. Zoom out. And again, it does look a little bit overwhelming and very long, But in some ways that shows the beauty of workflows, because you'd only have to make this sort of thing once and you could rerun it very easily. You wouldn't have to think about all of these steps every time. So it's quite good, I guess, to see the complexity can be captured here and repeated. So I won't go through what all of these steps are doing, but again, you can see that the main input here would be our variant calls, and that's going to go through lots and lots of steps, and it will eventually end, and we'll get consensus sequences for our samples. So let's click the home button again. Okay, so we'll run this workflow now. Go to workflow, consensus construction, the run button. So the inputs we need to give here, the reference genome, that's file number 2. The aligned reads, this is something we did, I think, in workflow 1. And this is file 136, which is the mapped read pairs. And again, this is discussed more in the tutorial if you want to know the reasons for some of these parameters and so on. So we're giving this a setting of 5 for the depth threshold. And then the variant calls, this is, in our case, file 637. So this is the final annotated variants. And the tutorial recommends just a slight change in these settings here. It recommends this at 0.7 and this at 0.25. So again, I won't run this now, but if you were going to, this is where you would click run workflow. And then all of those outputs would populate in your history at the top. So let's assume you have run that. And the main output here is our consensus sequences, file 1571. Let's have a look at that. Again, not very informative to sort of look at by eye, but you can see this is a multi-sample FASTA file with lots of sequences in it. One of the things we'll do next is try to see what lineages these different samples would be assigned to. There's various tools that you can do that with. The tutorial explains 2 of them. I'm just going to show one today. Scroll down. So we won't use the tool Pangolin today. We're just going to use the next one, which is Nextclade. So let's set this up to run now. What we would do in the tool panel is go to the phylogenetics section, click on phylogenetics, and then we can see Nextclade there. So click on Nextclade. We need to give Nextclade our consensus sequences, which was file that's our multi-sample consensus FASTA file, 1571. And here I want all the outputs. So I will select all of the outputs. And then if you were running this, then you would click execute. But I've already run that, so let's have a look at the output. So we have several outputs. The one that's probably most informative to look at is the TSV output. And so this is file 1591. Click on the eye icon. And it shows us what the different samples, what clades they've been assigned to. according to Nextclade. In the tutorial, there's a full description of all of those columns and what they mean. And there's a nice image of those clades in a phylogenetic format. So you can start to visualize how your samples are related. And so this sort of output from Galaxy is something you can explore more either within Galaxy or in different places with additional analyses. So as a summary then, here in this demonstration, we've run 3 workflows for SARS-CoV-2 data. Workflow 1 gave us those identified variants and some of the effects of those variants. Workflow 2 summarised all of that information over all of the samples and gave us a nice plot of the clustering of those samples and the genes and the mutations in different genes. And then workflow 3 took that information and characterised each of those samples into particular lineages. So I recommend, if you're keen to learn more about this, is repeating the tutorial, probably even step by step, is often a really good way to get your head around what the tools are doing and what the parameters are. And then if you're running this in any sort of scale, if you need to run it for lots of samples, that's when you move over to running with workflows to really speed up that analysis and to also really capture your analysis information so that you can make it reproducible. So this would capture all of the tools that you used, all of the tool versions, and all of the parameters. So it's a nice way to keep a record really clearly of what you've done. So Gareth, I think that's about all I was going to cover for the variant calling. Did you want to talk more about the workflow reporting?
Thanks, Anna. Yes, I'll take over for a little bit now. So to the audience, we're actually pulled back a little bit of time from where we were earlier today because we did take on the ambitious task that Anna delivered so well of showing hugely complicated workflows, quite frankly. And I really do want to echo Anna's point, even though she's one of our lead trainers, so I should be deferring to her. But please do step through these tutorials one by one. You'll learn an awful lot more about the tools. We've really shown you the work and good work and peer-reviewed work of ourselves and our global colleagues to construct these workflows. But ultimately, if you're analysing this kind of data, it is very worthwhile stepping through it step by step. We're not suggesting you iterate through all the settings of those tools, but to really understand what they do. And we're very sympathetic and don't want to sound elitist here, but as bioinformaticians, this is our bread and butter, to move files from one file format to another, to understand what the input file format is and what the tool settings are. So the workflows are there, they're digestible and they're there for you to use, but please do take that extra time to step through them to understand what they do. Galaxy has a failure language, so if a tool breaks, it will tell you, which is very useful. But sometimes just understanding the process is the best way to go. And before I share my screen, I guess on a closing note, these workflows, as Anna's shown, they're long and possibly one of the more frustrating things you'll experience, and I do hope you won't experience this, is setting up that workflow with hundreds of samples and having the second last tool of the workflow fail. This does not undermine the whole workflow, and there are ways to reinitiate just those elements of the workflow that have failed for one reason or another. But we would strongly recommend, if you're going to run these workflows, please do it on a small number of samples at first, real life samples, and build up to the size of resourcing and processing that you need, because that will give you the best experience of your data analysis. So we still have about 40 minutes. I think I'll consume another 10 or 15. So Idris, I will put you on notice after I've finished if you would like to make some closing remarks. But I want to show a more, I guess, I was going to say human-facing aspect, and that's wrong. So the workflows have some fantastic outputs, and particularly the little pop-up that Anna showed, where you can step through the genes and the samples you have and look at the variants, I think is fantastic. In our experience of delivering training in bioinformatics, I think one of the pieces of feedback we get, particularly from lab scientists, but I think it comes from everywhere, is the wow factor. Often the result of a lot of computational time is a text file. That's fantastic because there's your data, but it doesn't have you know, you might need to say things have worked well. So there's a relatively new feature in Galaxy at the moment. It's been out for a version or two, and it's still being heavily tuned on the service. And this feature is called workflow reports. And it's something I wanted to show to you today, just to give you, again, a taste of where you can take your data once it's computed. And I can see there's been a question in chat about the training material, so thank you, Anna, for addressing that. Yeah, so Anna, I'll leave you to just support that. Thank you. But we can always circle back to that later. So welcome to my Galaxy. I will just disable that video just to make sure we're all good. So Anna, to exemplify everything that we've been talking about today, shared with me her history. So here is the completed history for SARS-CoV-2 genome assembly from the earlier session today. And I don't think we had time to cover it earlier today, so apologies if we go over it. But you'll note when you look through this history in particular, files are tagged with styles of data. So the paired end data has actually a tag called Illumina, which allows you to, oops, not delete it, pardon me. This allows you to quickly navigate through your history to find those items that come from either a particular style of data, but you could easily imagine that this was any piece of metadata. It could be the sex of the individual, it could be a sampling site on the body, it could be were they COVID positive or negative, were they vaccinated or unvaccinated. So, and you can have multiple pieces of tagged data. These will propagate through your work. So any analysis you do on number 7 will propagate the Illumina tag through to number 12, to number 52, and so on. And this can be a fantastic way. I might just control that. So this is one of those really nice features in Galaxy that allows you to help navigate large pieces of data. And if you're watching closely during Anna's last work, I think there were something like 71 or shown items, and there are also over 1,000 hidden items. So tags are a great way to do that. But the feature I wanted to describe, like I say, is relatively new, and it is called pages or reports. They have 2 different names. I have created one here called APAN53. We're going to look at it first, then come back and understand what this report is. So here is a report I've created for today. I have, thank you very much, kindly borrowed the logo from the conference website, pardon me. I've given it a title, APAN53, and the title of the tutorial we followed by the URL. I've given it a timestamp, and now this timestamp is a bit more universal because it comes in at 10:23, which is not the time we're operating on right here. The version of Galaxy we analysed the data on And then a bandage image selected essentially at random from the history, but a bandage image from that history. Followed by a summary, a peek into the data set called summary numbers, where we can come through and look at the total sequences, filtered sequences, etc. The job metrics for one of the jobs. Now, I haven't picked a job associated with the above images. This is just to show you what went on. This job took 1 minute to run. It was given 16 CPUs. It was run here in Pawsey at our supercompute center, and it consumed 61 gig of memory. So all this can be put together very nicely for you and very quickly. And again, in the spirit of automation and reproducibility, this can be set up as part of your workflow, so it's auto-generated. So when you imagine a scenario where you are routinely passing SARS-CoV-2 data into Galaxy for processing, you would have a reporting template like this set up where you would have versions, dates run. You can actually add in sample names, and we'll do some of that now. So I quite like these. I think they're very approachable and they're shareable. So let's have a look. So this is what the workflow report looks like in the background. For those of you familiar with this, you'll note that this is essentially a markdown language. So it has a structure for insert objects. It has a structure for headings, subheadings, which I've made use of for free text fields. It isn't a full rich markdown language at this point. So I have had previous trouble colorizing particular words, for example, but I'm expecting this to come. Everything here on the screen is what is going to be called up. So there's our job metrics, our data sets, the bandage graph, the versions, etc. I feel like this could have more in it though. And so I want to show you how easy it is to add to the workflow report. And there's multiple ways of doing this. So the first is, here are all the possibilities that you can add to the workflow report. And you'll see that there are some really rich graphing options. And I'm not necessarily going to explore all these at the moment because they do just take a little bit more time to set up. And you also, for example, in the case of an XY scatter plot, need to be able to identify where in your data you have the style of data that would be applicable to that graph. But they're all in here and they can be added in. What I feel like is missing from this might be things like, what was the original data I analyzed? So I could either add the whole data set, and that might be a bit dangerous because I really just want to find out a little bit about the data. I don't necessarily need to embed all the data in the report. So I might say, what is the name of the data set I computed this on? And in fact, I want to see it straight before the image. I want to see this right up the top, and I'm going to say input data. It's not headed, and I'll give it the name of the data set, item number 7. Let's see if it allows me to do that because it is a collection. There is the collection. So there's our input data. I have only picked one, but I could pick multiple. And I just want to confirm what the data set type was because I know I have Illumina and Oxford Nanopore data. So I'm going to come down to the bottom again, page 2, and just find out what the data set type is for that particular data I'm computing against. I could add any other number of things in there. I'm going to come down here in a moment and look at workflows, but modifying this report is as easy as saving the report or saving and viewing, which is what I'm going to do now. My logo again, here's my report. Now here is my input data. I can see what I computed against. I can confirm the file type. That's looking pretty good. Let's add something else to the report and see how that looks. So I'm fairly happy with that. I'm going to come right down to the bottom here and I'm curious to see if I can add the workflow. Now, I did try to extract the workflow from this, so it should be somewhere in my menu. So let's just see if I can find it quickly. I'm not, where have all my workflows gone? It's terrible. They're all sorted by 2019. There it is. And I'm going to add that workflow in. In fact, I've actually added in a little bit lazy inside another image. So I'm just going to correct my formatting, take that out, and save and close. And here's our workflow listed down the bottom. So now I get a complete picture of what I have done to this job. I get a nice signature up the top of the company that helped perform it or the person I'm doing it for. The name, I could put my authorship in here, so I could put my name in here. Galaxy version, which will change if Galaxy changes the input data, the result I'm interested in, the result expressed as a table, job metrics if I want to know how long it ran, and the actual workflow I ran to achieve that work. So these workflow reports, I think, are fantastic. Like I say, they can be generated by a workflow. So I made that this afternoon, my afternoon, apologies, by doing add new page, and I based it on the data set that Anna had shared with me. You can also generate this out of workflows. So for every workflow you run, this will be recorded inside Galaxy. So here it shows that I ran a workflow 17 days ago. It has this particular name, and I can actually rerun that workflow if I want to. I can click on the workflow and find out where it's up to. So this is particularly useful if you're tracking workflows and you want to see which ones have started and finished. And if you're wondering what's happening here, this is polling the Galaxy database. It's pulling in all the jobs I ran on that workflow on that particular day. If there's a lot of them, that will be interrogating the database for some time. If there's a lot of steps, that will also take some time. I could go for a shorter workflow like this one here, but now I'm actually asking for 2 activities, so I'm probably just slowing the system up. I was hoping this would finish fairly quickly, because when this finishes, you'll see that you can actually find your workflow reports from this page. Now, looks like that is going to take a bit too long. But nonetheless, that is where you find them inside your Galaxy page. So all this history is here, and I can go back and access any data I've run before, see where it's up to, rerun it, or visualize it. So like I say, we are a little ahead of time. We've got about 25 minutes left, but I will unshare my screen. I'll also turn my camera on. Idris, if you do have any words you would like to say at the end, please do. Otherwise, I think we'll be wrapping up fairly soon.
Hi, everyone. I'm just amazed that we've got such a following still. I mean, this is a very long workshop, and yes, thank you so much, Anna and Gareth, for your persistence and incredible detail that you presented to us in each of those workshops you've had for the intermediate and advanced levels. I'm just here to kind of wrap it up to mention that we have a couple of more activities. That's our eResearch Southeast Asia activity. The one that's coming up on Wednesday, that is just coming up with a panel discussion on the accelerating the paradigm shift towards open science and data sharing in the pandemic times and beyond. This is pretty much geared towards non-specialists, but specialists are most welcome too to come to discuss open data, open access, open source, and also open peer review tools and platforms. We also will be discussing various funding sources and organizations that have been putting together facilities and resources. And we have amazing speakers ranging from people from university, that is, we have Professor Jeannie Balbo from University of Queensland and Open Access Australia. We have Julian Cribb, who is an author, but also former CSIRO National Awareness Campaign, public communications. We have two community leaders, one from India, Saranjit Kaur, who is leading the research software engineers Asia, and also Dr. Sandersan Oni, who is being active in the Southeast Asia open science community. So yes, that would be on Wednesday. On the Bangladesh UTC +6 time, it starts at 3:30 p.m., finishes at 5 p.m. And last but not least, we also have a discussion further on the Thursday as part of the Internet Access Management Group. We'll be discussing the Global Alliance for Genomics and Health Organizations tool that they've developed, but also has been implemented in Australia by Gareth and Anna's group, that is by Biocommons, Australian Biocommons, and has been implemented with a number of other health institutions. So yeah, that would be happening on Thursday, and it will be starting at 9 a.m. Bangladesh time, that's UTC +6, but I think our discussion will come last, closer to about around 11. The activity will continue until about 12 or 12-ish. So I was just wondering, was there any room for questions or any other sort of procedural matters that people might want to raise? I mean, let me see. Gareth, did you had any? I mean, the chat, and if you check the chat.
I think we've kept up with the chat, and thank you, Anna, for that. Yeah, so no, just thank you everyone for your attendance, and I hope this is allowed you to more of the do the analysis.
Right, thank you again, and you have all the other resources that will be put together after the conference. There will be a folder associated with this webinar spot. So any material that you might want to pass on to people, participants, they will be available on that folder. So yeah, I will hope that you know, people might want to come up with their comments or suggestions. Please let us know any particular query that you might have. So thank you very much again, everyone, for participating tonight. It's been a long day, but I'm sure that there's a lot there to absorb, and I hope you can join us with the other events, but also enjoy the rest of the conference. Thanks again. Bye now.